Good morning, everybody. We are going to start on chapter 6, which is microbial metabolism. So the term metabolism, some of all of our chemical reactions within a body cell. So in a microbe, that's some of all of their reactions because all microbes are single cellular. So I shouldn't say that. All prokaryotic microbes are single cellular organisms. So metabolism in a nutshell is the biological or biochemical reactions that take place inside of the cells of organisms. They can be broken into two basic types. Catabolism. So I always like to think of catabolism. It's a breakdown reaction, and if you've ever had a cat, you know cats jump on your counters and they knock glasses off, so they break things. A catabolic reaction is one that breaks substances down from larger to smaller subunits, and in the process, it releases energy. Anabolism, then, is the opposite reaction where you're building something up from smaller subunits, which usually means you need to take in energy. So you use energy to produce the end product. Here's an example of catabolic and anabolic reactions. In a catabolic reaction, we might break down glucose, and in that process, produce a bunch of energy. The energy that we use in the breakdown of glucose can be used to build up cell structures, make macromolecules, or even make the subunits to macromolecules that we need. There we go. There are two basic types of energy that we look at. Energy in a nutshell is the ability to do work. And the first type of energy is called potential energy. It's the energy that is stored. So potential energy is energy that's not being used yet, but can be used. Kinetic energy, then, is energy of movement. So the movement of water here, that's kinetic energy. Water standing still is potential energy. It has the ability to produce energy or to release energy, but it's not yet. The bonds in the food that you're eating or the drinks that you're drinking, they're potential energy. Once you break those bonds down, then they turn into kinetic energy. Do you know where all the energy in the universe comes from? Because we don't produce energy. We can't create energy, but we can change energy from one form to another. So the energy from the universe all comes from one basic source, the sun. That's right. So photosynthetic organisms harvest the sunlight energy, which is what drives all activities on Earth, and produces organic compounds. So it converts sunlight energy into the energy of food that they can use and we can use. Photosynthetic organisms then gain the majority of the energy on Earth, and then those organisms that eat photosynthetic organisms gain a small amount of their energy. This is called a 10% rule. Anytime you convert energy from one form to another, you lose about 90% of that energy. And in general, that energy that is lost as heat. And so only about 10% of the energy actually flows from one level to the next level in the ecosystems. So energy can be either stored or it can be released. Free energy is the energy that is usable to do work. So if energy is bound within bonds of a molecule, it's not considered free. But if we break those bonds, then it's considered free. Exergonic reactions are reactions where the reactants have more free energy, whereas endergonic reactions, the product has more free energy. So here, glucose has a lot more energy than its products. Do you see that? Glucose up here is a very high energy molecule. The products are low energy molecules. So we release energy as we break glucose down into carbon dioxide and water. That energy release can drive endergonic reactions. So here we have reactants that are very low in energy. The product is very high in energy. We need to supply energy to the reaction to build up that product. And this just comes from my anatomy and physiology textbook. I like this figure. It shows the exergonic reaction and the endergonic reaction. Typically, exergonic reactions drive endergonic reactions because we release energy, and then that can drive other reactions to occur. Enzymes are proteins that increase reaction rates by decreasing the amount of energy it takes for reactions to occur. Enzymes are called catalysts, and they basically work on every chemical reaction inside of cells. So we don't need enzymes to do their job to have a reaction occur, but for life to occur, we do. So if we're not living, then it doesn't really matter, but since we are alive, enzymatic reactions allow us to produce energy, to break things down, to build things up in a manner that is fast enough to keep us alive. Enzymes are reusable. They don't get used up in a reaction. They are very specific to their substrate. They're typically named for their substrate. They have a region called the active site where the substrate will bind. And you don't need a lot of enzymes for a reaction to occur because they work so rapidly. Some enzymes can catalyze millions of reactions every minute. And so enzymes work by binding the substrate to the active site. And that binding causes the enzyme to change in configuration just slightly. And then it can either build or stress bonds. So it can either build bonds or break bonds. And either produce multiple products or produce one larger end product, depending on the type of reaction we're looking at. Enzymes work by lowering the activation energy. That activation energy is the energy needed for a reaction to occur. And some enzymes work with coenzymes or cofactors. Cofactors are inorganic molecules that help enzymes work more effectively with their substrate. Coenzymes are organic molecules like vitamins that help enzymes and substrates work together. And enzymes work in what we call metabolic pathways. So a metabolic pathway is a very branched pathway. We don't just have one enzyme catalyzing a reaction and then we have the end product and then we're done. In general, we have multiple enzymes in a pathway, and then these enzymes produce products or intermediates that can move into multiple different paths. And so these are very branched. Metabolic pathways are very complex. This makes it look very nice and smooth. Here we have an enzyme and we have a substrate. The substrate binds to the enzyme. We produce some intermediate. That substrate binds to a second enzyme. We produce another intermediate. That substrate binds to another enzyme and we produce a final product. It's not really that pretty. It's more like this intermediate here may go over here or over here or up here or up here, and we produce lots of different products from one intermediate, okay? But I will say I like this figure because it shows enzyme inhibition, which we'll talk about. So there's competitive inhibition here, and there's non-competitive inhibition here, okay?
Organisms contain many different kinds of enzymes that catalyze a variety of different reactions. Many of these reactions, such as those involved in the biosynthesis of an amino acid, are carried out in a specific sequence called a biochemical pathway. In such pathways, a substrate is converted into a product by the first enzyme in the pathway, and the product of the first reaction then becomes the substrate for the next reaction. The product of the first reaction then becomes the substrate for the second enzyme. The sequence of reactions continues until the final product is made. When a biochemical pathway is functioning, the initial substrate is continually converted to the final product through the series of steps in the pathway.
Okay. So biochemical pathways are really important. They are going to allow us to produce end products that we need, but we don't need to use energy to produce end products if we don't need those end products. And so we do have inhibitions to biochemical pathways. Some of these inhibitions are going to stop pathways so that we don't waste energy. And sometimes we have inhibitors that block pathways because they are trying to kill off the organism, so they're not beneficial. Competitive inhibitors inhibit the substrate from binding to the active site. They basically compete for that active site. And oftentimes, competitive inhibitors are considered poisons and can kill the organism. Non-competitive inhibition is often how we control pathways when we don't need those pathways running anymore. And so a non-competitive inhibitor binds to a different region called an allosteric region. And that changes the shape of the active site so the substrate can't bind. And so here we see the active site, and there's the allosteric site. This substrate can bind to the active site if there's nothing bound in the allosteric region. But if we have an allosteric inhibitor, then the substrate can no longer bind. So this is often important when we're trying to control a pathway because we don't need any more end product. If, say, we're producing some amino acid and we've produced a lot of that amino acid, we don't need more of that amino acid, so we don't need this pathway to run. One of the amino acids can bind to the allosteric region and act as an inhibitor so that the substrate is not converted to more amino acid. That would be an example for why you would shut a pathway down using allosteric inhibition. Competitive inhibition then is where you have a competitor for the same active site. And so in this case, we're looking at a chemical that binds to a region on this enzyme. This is called a sulfa inhibitor. The sulfa inhibitor kind of resembles the actual substrate, and so when it binds, it doesn't allow the substrate to bind. And this can stop bacteria from growing and can protect the organism, the human, from whatever bacterial pathogen this would be. So enzymes are really important in metabolic pathways, but we also need energy for metabolic pathways. The energy we use is called ATP. ATP is adenosine triphosphate. So this is what it looks like. It's a nucleotide, which is found in the nucleic acid region. So we have this adenine nitrogenous base. We have a ribose sugar, and we have 3 phosphates. Adenine and the ribose make adenosine. 3 phosphates make it a triphosphate. This is what almost all cells use for energy, for movement, protein synthesis, other anabolic or catabolic reactions, cell division, everything. We use ATP. And when we break ATP down into adenosine diphosphate, or this adenosine molecule plus 2 phosphates, we release the energy, and that energy can then build up other substances or be used to break down molecules, whatever we need to use it for. The most common molecule we use for producing ATP is glucose. Glucose is a carbohydrate. It's a monosaccharide. In animal cells, we store it as glycogen. In other cells, like plant cells, they store it as starch. And then bacteria don't really store glucose as larger molecules. They just have glucose in their system if they're going to use glucose. So the breakdown of glucose releases ATP slowly, and it goes through multiple paths, metabolic pathways. The process is known as cellular respiration, and it's how cells break down glucose to produce energy. If it's an aerobic process, it's called aerobic cellular respiration. If it's anaerobic, it's called anaerobic respiration. So we're going to focus on aerobic cell respiration, which you probably learned at least once in Gen Bio if you took that, and in anatomy and physiology if you've learned that. Now we're going to talk about it in microbiology. We're going to go through it rather quickly, but we're going to look at the differences between bacterial and eukaryotic cells. So this shows the steps of cell respiration, and so for the good news is you don't have to know all of these steps. What I do expect you to know are what happens. So in glycolysis or glycolysis, which is the first step of the breakdown of glucose, we take a 6-carbon glucose molecule and break it into 3-carbon pyruvate molecules. And so you can see here's glucose. Glucose is activated using ATP to produce this this fructose 1,6-biphosphate, which is immediately broken down into 2 3-carbon glyceraldehyde 3-phosphates. And then it's oxidized, and and we remove phosphates from it. So when we oxidize it, we reduce something else. So here we have NAD that is oxidized to NADH, or that oxidizes the molecule. So NAD is reduced to NADH. Glucose was oxidized, and we produce a little bit of ATP. The production of ATP is through a process called substrate level phosphorylation. Substrate level phosphorylation is a very, very large term that just means we took a phosphate from one molecule and we added it to another molecule called ADP, adenosine diphosphate, producing ATP. And so we end up, once we've oxidized the glucose molecule, we end up with pyruvate. We end up with some NADH, which has been reduced and moves to the electron transport chain, and we produce a little bit of ATP, a total net gain of 2 ATP. This is a song that you can listen to. It's the Glycolysis song. It's a fun song. I like it. From here, we move into the cell, or well, depending if we're in bacteria, the glycolytic pathway occurs in the cytoplasm. If we're in a eukaryotic cell, the glycolytic pathway also occurs within the cytoplasm. If we're in bacteria, then transition stays in the cytoplasm and moves to the cell membrane. Whereas in a eukaryotic cell, transition occurs inside of the organelle called the mitochondria in a region known as the matrix, so inside the second or the inner membrane. So transition step is a step where we take pyruvate, which is a 3-carbon molecule, and break it down to a 2-carbon acetyl group. The acetyl group then is going to attach to coenzyme A and be brought to the citric acid cycle. So to convert pyruvate into acetyl coenzyme A, we're going to remove carbons. So pyruvate is a 3-carbon molecule. We're going to remove a carbon off of it, add it to oxygen, producing carbon dioxide, which is a waste product. And then we're going to oxidize the pyruvate, which means we have to reduce more NAD. The NADH that is produced in this process is going to move to the electron transport chain. Nope, I guess I don't. Oh, that's an easier picture, but we're going to look at this picture. This is your figure that shows the citric acid cycle. So the citric acid cycle, also known as the Krebs cycle, is a cycle that starts with a 4-carbon molecule called oxaloacetate, and it ends with that 4-carbon molecule called oxaloacetate. So we are producing, we start with and we end with the same molecule. The citric acid cycle occurs in the plasma membrane of the bacterial cell, or if we're in the eukaryotic cell, it occurs in the matrix of the mitochondria, so right next to the transition step. It's called citric acid cycle because we produce a citrate molecule. You can see that right here, citric acid. It's called the Krebs cycle by others because that's who found this pathway. So we start with the oxaloacetate. The acetyl group is dropped off by coenzyme A, and the acetyl group attaches to the oxaloacetate, producing the citric acid molecule, or the citrate. The citrate molecule then is going to be oxidized completely. So it's converted, but we're not going to worry about the conversions. It's going to be oxidized completely by NAD and by FAD now. These molecules are going to be reduced. And in this process, substrate level phosphorylation occurs, so we produce a little bit of ATP. For each acetyl group that enters, we're going to oxidize it completely, so we're going to produce a total of 2 carbon dioxides and 3 NADH and 1 FADH and 1 ATP. So because we have 2 acetyl groups going in for every glucose molecule that we break down, we have a total of 4 carbon dioxides, 2 ATP, 6 NADH, and 2 FADH. So that's what this is showing. And this is just a prettier picture. I mean, it's just simpler to look at. This is showing with each glucose that enters, we lose a carbon here. Or with each acetyl group that enters, we lose 1 carbon here, one hydrogen and electron there, 1 more carbon, another hydrogen electron. We produce ATP, we lose another hydrogen electron and another hydrogen electron. There are 2 acetyl groups that enter, so we produce a total of 4 CO2, 2 ATP, 6 NADH, and 2 FADH.